All right. Now, after the mast cell and everything that we saw in the previous video, we see that the tissue, the phagocytes, which are the tissue macrophages and the dendritic cells, detect what is going on, or the bacteria or whatever has come in. And there is also an increase, there's an increase in the permeability to monocytes, neutrophils that can easily pass through the vessel walls. And the monocytes are the ones that become the phagocytic macrophage, and they uh, migrate to the tissue. So what happens? What's the phagocytotic mechanism that happens here? So what you see is, first of all, we said the pathogens themselves have got proteins on them. But the first thing that we see here is the pathogens are chemically attracted to the phagocytes by a process known as chemotaxis. So chemotaxis, this is attraction of the pathogen to the phagocyte. So how does this happen? The chemotaxic property is C5a. All right, so this is what makes this possible. It makes it possible for the pathogen to be attracted to the phagocyte. All right, what's the next thing? The next thing we want to know is that the pathogen has C3b, okay, C3b proteins on its surface. All right, so it's being attracted to the phagocyte by C5As. All right, and this is known as chemotaxis. And then it has C3b on its uh, surface. So these C3b proteins are able to bind on C3b receptors. And guess who has C3b receptors? Exactly, the pathogen. So the pathogen has got C3b receptors. All right, let's see if we can draw them. So these C3b proteins that are found on the surface of the pathogen are able to bind to the C3b receptors on the phagocyte. All right. So let us start. First of all, we have C5a, chemotaxis, pulling or attracting the pathogen to the phagocyte. Okay? And the C3b proteins are then able to bind. Now, this binding or this coating of the pathogen uh, with the phagocyte receptor is known as opsonization. Now, in opsonization, we are looking at our pathogen and our phagocyte binding. All right, so let's see how we can do that. See if we can put it here. So it literally coats this pathogen. So if these are the C3Bs, then we have the phagocyte coating them or binding with them. All right. So upon binding, right, what is going to happen? Then we're going to see that it's going to be ingested. It's going to be taken into the phagocyte. So they carry the pathogen into, or because it's bound into, it will just be taken into the phagocyte. And in the phagocyte, it is kept in a phagosome. All right. So inside there, we see that it goes into the phagosome. Okay. And in the phagosome, that is where now we are going to find lysis material. Okay. So we find that it's going to be lysed. Okay, it's going to be lysed. Let me see if I can put something there. It's going to be broken down. Okay, so let's just see where we are right now. It's attracted, C5a, to the phagocyte. All right, then the C3b binds the C3b receptors. Okay, then the organism is engulfed into a phagosome. All right, and then the phagosome has lysis-like material. All right, so it breaks down this pathogen. Okay, so what happens after breaking down? So let's see. It breaks down. It breaks down the pathogen. So when the organism is killed, what then happens? So it's killed, it's digested, broken down. You find that then the content, the protein content of this pathogen, some of it is presented on the surface of the phagocyte. Okay? So some of it is presented on the surface. What is presented? The protein. The protein of what? The pathogen. To say, yes, we got rid of this guy. And specifically, this is the guy that we got rid of. And the rest of it is exocytized. Exocytized. Whew. Yeah. Exocytized. Exocytized. It's taken out. All right, so what do we really mean when we say antigen presentation? So this is when we have the invading foreign organisms that are engulfed. Okay, so we're talking about macrophages. They're the ones that have an ability to do this, and then or dendritic cells. So later, the antigen from these organisms is then digested, and small peptide products from this one, like we said here, are placed on the surface of the phagocyte. All right. So these antigenic peptide products are specifically placed on the MHC. Remember this guy? Major histocompatibility 2. Remember I talked about 1, but in this case it is placed on the MHC2. And this is situated on the surface of antigen-presenting cells. So these include B cells, okay, and other cells that are able to present the antigens. So once they present these antigens, then we have the next step coming up. So what have we talked about in this video? In this video, we have talked about how that the pathogen is attracted to the phagocyte and how it is ingested, lysis happens, and that it is removed after that. But most above all, we've talked about how that there's presentation of this of the some peptides of this pathogen on the surface of the phagocyte, specifically on the MHC2. In the next one, we will talk about what happens next. Yes, it has been presented. What next?